Most people are completely unaware of the complex communities of small-bodied organisms that exist all around them. However, these microcommunities are important parts of a diverse ecosystem and lead to greater understanding of biodiversity. The microcommunity formed by the gall wasp Discocaspis cinerosa on the Texas live oak is an important part of global biodiversity. Galls are abnormal growths of plant tissue induced by the activities of bacteria, fungi, and insects. Insect galls are formed to house larvae and serve as a way to protect larvae for later emergence. The mechanism that causes the formation of galls by D. cinerosa wasps is not well known. The galls induce production of nectar, as seen in the photo, which attracts ants. The ants, in return, protect the developing wasp from predation by larger predatory wasps. The galls dry up in the winter after larvae emerge and turn black, as seen in the background image. Afterwards, the dry galls become a home for many other species. Since the system is very poorly understood, I sought to study the relationship between the presence of these galls and the affected status of neighboring trees. I also attempted to determine whether or not the number of acorns was reduced by the gall presence. Data was collected in transects at the UT main campus, in Hyde Park, and at Breckenridge Field Lab. A regression of the acorn data showed that there was no relationship between the number of galls on a tree and the number of acorns produced. As expected, there was a significant relationship between the gall status of the tree of study and its closest neighbor. 82% of infected trees also had infected neighbors. The clear relationship between gall presence and neighborship suggests that branch-to-branch transference may be the path of gall infection. Canopy-based studies of the phenomenon may lead to more insight. A lack of a relationship between galls and reproductive ability was expected due to past research, but the result may have also been caused by a small sample size. Larger studies that span several seasons of gall formation and death may give insight into the variation of gall density. Localized studies of individual branches may also show the spread. Future personal research will address the role of ants in gall distribution, as well as the potential acquisition of immunity to galls, as reported by Texas A&M researchers. Hopefully, future work to address biodiversity will focus on the perhaps less charismatic fauna and flora